name is Bristena Oprisanu, and I'm going to talk to you about our paper on utility and privacy in synthetic genomic data. With the cost of genome sequencing falling at a rate much faster than Moore's law, more and more projects focusing on genome sequencing are emerging, such as the Yale Cancer Center scientists built Genomic research platform to help treat cervical cancer, Singapore researchers create the world's largest Asian genetic data bank, or the National Institute of Health backs a new 7 million genome center for the All of Us research program. However, as more and more genomic data is generated, collected, and shared, serious privacy, security, and ethical concerns become relevant. And we have seen several attacks and privacy concerns in the media, such as the NHS service exposed thousands of client records online, or the NHS patients' genetic data targeted as foreign hackers attack high security Ministry of Defense unit, or that China uses DNA to track its people, and several others. Now the question is there a solution to help with this privacy concern? And in many cases, people have come up with the answer of synthetic data. And in fact, synthetic data has been proposed in healthcare as a way to widen data sharing without the loss of privacy for patients, as, for example, is in the case of the NHS England, which has a pilot program for accidents and emergency synthetic data. So what is synthetic data? First of all, synthetic data is mostly created using generative models, and a generative model is a statistical model that, given a raw data set, learns an approximate distribution of the data, and from that trained model, we can then generate a synthetic data set by sampling from the learned distribution. The promise of synthetic data is that we can conduct statistical analysis on the synthetic data, which will lead the same insights as an analysis conducted on the original data. And at the same time, for the sake of the argument, because there is no direct linkage between the synthetic and raw data, privacy adversaries cannot exploit this knowledge. In this work, we conduct a measurement study on generative models purposely built for genomic data, with the aim of answering the question of whether this synthetic data is indeed the silver bullet that people think. For this purpose, we chose three data sets, two smaller data sets from the HapMap project and one larger data set from the 1000 Genomes project, as well as several generative models. The first one is the recombination model, which is based on a high-order SNV correlation that relates the linkage disequilibrium patterns in the underlying recombination rate, and has been proposed by Samani et al. Then we used two models proposed by Yelman et al. In their paper Creating Artificial Human Genomes Using Generative Models. We also choose a WGAN model proposed by Catamaran et al. However, the two HapMap data sets specifically, they're quite small, so in order to overcome issues that may be caused by low number of training samples, we also proposed two hybrid approaches, namely Recomb RBM and Recombination GAN, where we use the former to generate extra samples, which we then use together with the real data samples to train the RBM or GAN models with the same parameters as before. So now let's look at some utility statistics, and the first one is going to be the major allele frequency, or MAF, which is routinely used to provide helpful information to differentiate between common and rare variants in the population. And for the CEU dataset, we observed that from a visual inspection that the Recomb and the WGAN are the ones that replicate best the real data, and is the same case for the CHB. Whereas when we look at the 1000 Genome project, again, the Recomb maps distribution is also similar to the real data. However, the RBM and RecRBM both display maps closer to the real data. We also did a non-parametric statistical significance test, namely the KS2 sample test for all data sets, and found that for the CEU and CHB population, we cannot reject the null hypothesis only for the samples generated by Recomb and WGAN, whereas for the 1000 genomes, we cannot reject the null hypothesis for Recomb, RBM, and RecRBM. Moving on, we look at the percentage of heterozygous samples in each of the data sets. And this is commonly used in population statistics, as a low percentage of heterozygote variants imply less diversity in the population. So here we plot the percentage of heterozygote variants in each sample for the CU and CHB population, comparing the real statistics, which are the blue leftmost bars, with those computed on the synthetic data. In both cases, RECOMB and WGAN yield similar percentages to the real data sets. And when we compute the KS statistics in this case, we found that we cannot reject the null hypothesis for RECOMB and WGAN in the CEU datasets. However, we actually reject the null hypothesis for all synthetic datasets generated by from the CHB dataset. With the 1000 genomes, the percentage of heterozygote samples in the real data is lower across all samples. And actually, we find that the GAN and the REC-GAN significantly deviate from the percentage of heterozygote samples found in the real data. So in the bottom plot, we remove those two models in order to observe better the other percentages. And for this, in this case, for the 1000 genomes dataset, we do not reject the null hypothesis, only for the RBM. The next statistics we look at is the linkage disequilibrium, which captures the non-random association of alleles at 2 or more position in the general population. That means that those alleles do not occur randomly with respect to each other. And it has a value between 0 and 1, where 0 means there is no LD between the two SNPs, and 1 means that the SNPs are in complete LD. For the CEU and CHB population, RBM generates samples that display a stronger LD than the real data, with more training samples. REC-RBM yields a weaker LD, but still stronger than the real data for both models. On the other side of the spectrum, for the REC-GAN, the LD for the synthetic data is the weakest. Now, looking at the bottom plot for the 1000 genomes, we found that a stronger LD is between the real samples than for the other two datasets, and that RBM generates samples that are almost indistinguishable from the real data in terms of LD. We find in this case that we cannot reject the null hypothesis for samples generated by RECOMB, GAN, and WGAN, and which would imply that even though the GAN models did not preserve well allele frequencies or population statistics, it manages to preserve the correlations between allele. Now, for the 1000 genome dataset, the only generative models that yield samples which cannot be rejected under the null hypothesis is the RBM. Now, we look at some privacy. We look at some privacy threats, and more specifically, we look at membership inference attacks, since it's one of the best understood privacy threats in both genomics and machine learning. And it is inherently the ability to infer the presence of an individual's data in a data set within a data set. And for this, in order to implement a membership inference attack, we use a shadow modeling technique, which was originally introduced by Chokri et al. And what we do is we take a reference data set, which we split a subsample into multiple data sets. Then we take a target, which we add to those data sets. And for each of those data sets, with or without the target, we train generative models and generate synthetic data sets, which we label as in or out, depending on whether the data set from which the synthetic data set was generated contained the target or not. We do this for all our subsamples. And then we use these synthetic data sets in order to train a membership classifier, which, given a synthetic data set, will then be able to tell us whether the target was present or not in the raw data set that generated this synthetic data. So for this, we use the metric of privacy gain, which will range between 0 and 0.5, where a privacy gain of 0 means that publishing the synthetic data set is equivalent to publishing the real data set. A privacy gain of 0.25 means that the adversary has no advantage over random guessing when publishing the synthetic data set. And finally, a privacy gain of 0.5 means that publishing the synthetic data set reduces the adversary's chance of success. Now let's look at the privacy gain for the 2-half data set. And what we find is that we have a low predictability of privacy gain without fixing the target and the training data set first. And for a good privacy mechanism, we actually need the privacy gain to be predictable, which is not the case. So actually we found that in the case of logistic regression, when we use the histogram feature extraction techniques, for example, for the RECOMB model, which had the best utility statistics, we found that 94% of the targets have a privacy gain below the 0.25. And for the same feature set, in the case of the RBM, we found that 96% of the targets have a privacy gain below 0.25, which is the random guess baseline. Now, for the 1000 genomes, we only looked at the RBM since it was the model that consistently performed better than the others. And we found, like similarly to the HapMap case, that privacy is not really predictable, suggesting that just having synthetic data sets is not a good privacy mechanism. However, the other interesting case that we did for the 1000 genomes was to which we did in order to better understand whether or not, with more training data, a target signal in the synthetic data set is diluted. We tested the case of an extreme outlier. That is, we crafted an outlier that only has minor alleles at all positions. And while we are aware that this is not really possible in a real-world scenario, our goal was just to observe whether and how much this impacts the privacy gain. We tested this across 10 test runs, and surprisingly, we found that now the privacy gain is centered around 0.25, which was the random guess baseline, which might suggest that the RBM model actually kills outliers when generating the synthetic data. Now, we also implemented a new privacy membership inference attack, where only partial information was given to the attacker. So compared to the previous attack, we only give the attacker access to a fraction of SMVs from the target sequence chosen at random. And now the attacker will use the recombination model as an inference method to predict the rest of the sequence. We also adjust the privacy gain formula. Now, looking at the recombination model, we actually find that even with only 0.05 of the target SMVs available to the attacker, for 90% of the targets from the histogram feature set and 91% of the targets from the ensemble feature set, the attacker's accuracy is still above the random guess baseline. And our intuition is that many targets are vulnerable to this attack, even with partial information, since we are using the recombination model not only for the attack, but also as an inference method. Now, we also implemented this membership inference attack with partial information for the RBM. And across all feature sets, in this case, there is an increase in the accuracy of the attack with more information available, which is unsurprising. And if we look at the CDF of the privacy gain in the case of partial information available to the attacker, we found that under the naive feature set, increasing the partial information available to the attacker actually negatively correlates with the percentage of targets which have a negative privacy gain. And again, under all feature sets, for most targets, releasing the synthetic data set instead of the real data yields a positive privacy gain, meaning that releasing the synthetic data set instead of the real data sets improves the privacy gain. Overall, not even with partial data from the target sequence, we don't obtain privacy gain values consistently better than random guessing, which, as mentioned before, indicates that synthetic data is not really a reliable privacy defense. In conclusion, we find that the different combinations of data sets, attack classifiers, and feature sets yield varied results with respect to privacy. This is due to two main reasons. First, not all classifiers have the same accuracy on tasks for the same data sets, as shown in previous work. And secondly, the features that the generative model preserves after training will reflect in the synthetic data. Thus, it will impact the privacy gain based on the feature extraction method. Then we also saw that high-quality synthetic data must accurately capture the relations between data points. However, this can enable attackers to infer sensitive information about the training data used to generate the synthetic data. And this was in fact illustrated by the performance of the recom model on the HapMap dataset. While it achieves the best utility, it does so at a cost of significantly reducing privacy. We also found that the size of the training dataset matters, especially in the case of non-statistical generative models. However, overall, we did not find any single method that outperforms all the others for all metrics and dataset. Thank you very much, and I'm happy to take questions.